Welcome viewers. Today we will be discussing on the topic Basics of Molecular Biology. Molecular biology is the study of gene replication, transcription and translation. Herein, the main components of molecular biology are DNA, RNA and proteins. If we look into the cell, we will see that it comprises of chromosome. Inside the chromosome, there exists the genes. Genes are the stretches of DNA that are going to code for proteins. Let's have a look onto the DNA double helix. It comprises of bases adenine, guanine, thymine and cytosine, which are forming hydrogen bonds, and the structure was given by Watson and Crick. Next important component is RNA. Unlike DNA, it is single-stranded, and herein we will see the existence of uracil, which was not present in DNA, wherein thymine was present. Next, let's have a comparative look on the structure of DNA and RNA. Herein you can see the double helical structure of DNA and the single-stranded structure of RNA, wherein the nitrogenous base thymine is being replaced by uracil in RNA. In few cases, we can see the existence of double-stranded RNA, not in case of bacteria, but in case of certain viruses. Next, the important three types of RNA are mRNA, which stands for messenger RNA, rRNA, which stands for ribosomal RNA, and tRNA, that stands for transfer RNA. They play a vital role in the process of translation. What is the function of mRNA? mRNA comprises of the message that is going to be conveyed in terms of sequence of amino acids, which finally leads to the formation of a protein. What is the function of tRNA? tRNA acts as an adapter molecule, as it has got amino acids attached to it, and it also is capable of interacting with the codons of the mRNA. Third important component is the ribosomal RNA. Ribosomal RNA are the parts of ribosomes, which is the site wherein the real process of translation takes place. Let's start the journey of replication. In brief, you will see that how the process takes place and what are the important components of DNA replication. You will see that DNA polymerase is responsible for carrying on the process of replication. Herein, there is involvement of an RNA primer, which is responsible for priming and initiating the process of DNA replication. Herein, we will see that there are two types of fragments. One is that is synthesized continuously, and second strand is synthesized discontinuously, and that leads to the formation of Okazaki fragments. Herein, we will see the involvement of enzymes like helicases, single-stranded binding proteins and topoisomerases. They are together working to carry on the process of DNA replication. So, summary of the replication in brief is that helicases will unwind the parental double helix. Next step will be single-stranded binding proteins will stabilize the unwound parental strand. And the third step, that the leading strand is synthesized continuously in the direction of 5' to 3'. And the lagging strand is synthesized, as I said, discontinuously, and these fragments are called Okazaki fragments. And finally, after the primer is replaced by DNA by another DNA polymerase, and these ends are joined together with the help of enzyme called ligases. Another important process called gene transcription. The components which are involved in the process of gene transcription are the double-stranded DNA, out of which only one strand participates in the process of transcription at a time. The sequences that play a vital role are the promoter sequences, which are being recognized by RNA polymerase the enzyme which is responsible for carrying on the process of transcription. And transcription starts from a region called the start site or start signal. And as transcription is complete, it terminates at a site called transcription termination site. They may be row dependent or row independent transcription termination signals, which is basically the existence of hairpin structures and a stretch of poly-U residues in the RNA. Another important aspect to be studied in molecular biology is the reverse transcription process. As I said, the transcription is conversion of DNA into RNA. So obviously, reverse transcription will be conversion of RNA to DNA. This thing actually happens inside the retroviruses, as because retroviruses do have got RNA genomes, and they introduce their RNA genomes inside their host cells. Inside those cells, the reverse transcription process, that is the conversion of RNA to DNA, takes place, taking help of the enzyme called reverse transcriptase. After that, the DNA is getting transcribed and translated to produce important proteins which are involved in the replication of the genome as well as packaging of this viral genome. So you can see here the retrovirus, how it infects. It is getting inside the cell through endocytosis. Then the viral RNA is undergoing reverse transcription and it gets integrated inside the host genome, or it may have its independent existence. If it has independent existence, then it will carry on the transcription of its genome and will synthesize proteins which are involved in packaging of their genomes, which will help them in their process of coming out of the cell by the process called budding. Now we are coming to another important aspect of molecular biology, that is the genetic code. As because we all know that DNA is made up of 4 important components, that is adenine, thymine, guanine and cytosine. On the other hand, the protein comprises of 20 essential amino acids. So it is the job of these 4 different nucleotides to code for 20 different amino acids. And this problem was solved by the concept of genetic code, which states that 3 nucleotides will be coding for 1 amino acid. This leads to the existence of 64 different codons, out of which 61 are coding for amino acids and 3 different codons are existing and which are working as stop codons to stop the process of translation. Francis Crick, who in the year 1966 gave the concept of Wobble. Wobble hypothesis states that 1 amino acid can be coded by more than 1 codons, and if it is so, then it should follow certain rules. Let's have a look on those rules. It states that if the first anticodon base is G, it can pair with C as usual, or in a Wobble mode, it can pair with uracil. For example, tRNA for histidine with GUG as anticodon can recognize both CAC and CAU as codons. Next rule is, if first anticodon base is U, it can pair with A as well as G. Whenever an amino acid is encoded by a pair of codons, the third codon bases are U and C. The process of translation, which is the conversion of the message coded in the mRNA into a polymer of amino acids or proteins. Here, the important components are mRNA, tRNA and ribosomes, which comprises of ribosomal RNA and proteins. So you can see that this synthesis also takes place in the direction 5' to 3'. Herein, the steps of translation are summarized as the ribosome has got 3 sites. One is called P site for peptidyl tRNA binding site, A site called aminoacyl tRNA binding site, and E site is called as the exit site. So initially the formyl methionine tRNA or initiated tRNA will bind to the P site, and after that the other amino acid bearing tRNAs will be entering through the A site. The second step will be the formation of a peptide bond between the amino acids which are residing in the A site as well as the amino acid which is residing in the P site. Once peptide bond formation takes place, this leads to the movement of the ribosome one step ahead, and the result of it is that there is generation of a new vacant A site and the uncharged tRNA is moving on to the E site. Then we will see a fresh lot of charged tRNA will be entering the A site again, and the process continues until and unless the A site reaches the stop codons, for which we don't have any tRNAs, and then there will come release factors to terminate the process of translation. Tools which are used in molecular biology. Number one, and the most important, is the gel electrophoresis, wherein the macromolecules are separated on the basis of their sizes and charges. There are various types of gel electrophoresis systems. We have got agarose gel electrophoresis systems to separate DNA. We have got polyacrylamide gel electrophoresis systems to separate out the proteins. In agarose gel electrophoresis, as you can see here, that agarose is melted and the gel is casted, and you can see the nature of the gel. After the gel solidifies, we put it inside a buffer tank. After that, we will be loading it with DNA, and to track the existence of DNA, we have added a bromophenol blue as the tracking dye. And after running of the gel, the separation of the macromolecule DNA is visualized by putting the gel on an UV transilluminator, right? And the second, as you can see here, is sodium dodecyl sulfate polyacrylamide gel electrophoresis apparatus. This is used for the separation of proteins, and the gel is cast by the polymerization of acrylamide and bisacrylamide. You can see here that how the gels are running, and they are getting separated on the basis of their molecular weights. There is another method wherein we can separate proteins on the basis of their charges as well as their molecular weight. Such a gel is called as two-dimensional gel electrophoresis. The second important field of molecular biology is PCR, that is polymerase chain reaction, and this is supposed to be one of the most important discoveries of 20th century, where scientist Carey Mullis in the year 1984 discovered this methodology, and for that he was awarded Nobel Prize. This is an in vitro method of amplification of DNA. You can see here that exponentially how you can generate DNA just in a very feeble amount of time. It comprises of steps, namely denaturation for separation of the strands, annealing for annealing of the primers onto the templates, and extension, that is polymerization of the DNA. This is a method called cell-free cloning, or we can amplify the DNA in an in vitro method. The major applications of PCR are: we can study the patterns of gene expression using PCR technology. The task of DNA sequencing is made tremendously easy with the development of PCR. PCR has numerous applications to the more traditional process of DNA cloning, and one of the exciting applications of PCR is the phylogenetic analysis of DNA from ancient sources. And another important application of PCR is the pattern of genetic mapping. It also helps us in paternity detection assays or determination of various criminals from by collection of DNA or collection of blood samples from the crime spots. Now, we we are coming to another important aspect of molecular biology, that is the macromolecule blotting and probing. It comprises of northern blot, wherein the gene expression can be studied, and herein we will separate the RNA on an agarose gel and we will be blotting it with a radiolabeled or some probes. This will help us to check whether the gene expression is occurring or not. Then we do have southern blotting, wherein we can see that we will be separating the DNA and we will be blotting it to get the presence of a particular gene or not. Say for example, you are working on a new bacteria and you want to check that whether it has got lac operon on or not. Then you can use southern blotting to check the presence of LAC-Z gene. Similarly, another method is western blotting, wherein we can see that it helps us to determine the presence of a protein or not. Here, the protein molecules are separated on a polyacrylamide gel, and after that they are transferred onto a nylon membrane or PVDF membrane, and after transferring it onto the membrane, we are blotting with a antibody which is specific to that particular protein. Then we will be adding secondary antibodies tagged with certain enzymes. These secondary antibodies will bind to the Fc portion of the primary antibody. If we add the corresponding substrate, then the enzymes will act on it and we will get a colored reaction. So you can see here the expression of a gene and the existence of the protein. All this can be studied taking help of southern blot, northern blot and western blot respectively. Molecular markers are being used extensively to study different organisms and to establish their relatedness. So, most important markers that are used are methods are RFLP, called restriction fragment length polymorphism, wherein the DNA is digested with restriction endonucleases and different patterns are being obtained. Say for example, herein you can see that the parents' DNA are being digested and the patterns are being studied, and thereby it can be estimated that what will be the patterns of their siblings. Second important method is AFLP, or amplified fragment length polymorphism. Herein we will be using the method of PCR, wherein specific primers are used and the regions are being amplified and they are separated on a gel electrophoresis method. Herein we will see that there are certain repeat sequences and the number of repeats varies from individual to individual. So, this repeats are varying, so the amplicon length will vary and each length will be a significant marker of a particular individual. These things are being maintained to establish many criminals or to understand the persons who are involved in criminal cases and paternity detection methods. Third important method is RAPD, or random amplification of polymorphic DNA. Here, very short stretches of primers, say of 10 nucleotides of length, random primers are used and it is used to amplify DNA using the genomic DNA. We will be getting a pattern of profiles which are being run on an agarose gel and they are compared to understand the relatedness among the organisms. Say for example, if I want to compare different bacteria isolated from a particular site and we have got no information about them, then we will be using this RAPD method. Next methods that are used as simple sequence repeats. Here also we will be designing primers and we will be amplifying it and we will be studying their patterns. Another important methodology is called single nucleotide polymorphism, or SNPs. SNPs are used nowadays have got a major role to play, as because different individuals may have a change in the nucleotides. As you can see here that in individual 1 we have got A and individual 2 has got G in its place and individual 3 has got T in this place. So just by having the SNPs, we can determine or differentiate between the individuals. In our today's lecture, we have gone through the important aspects of molecular biology. We have seen that the three important components of molecular biology are DNA, RNA and proteins, and how the DNA replication takes place and how the gene gets transcribed and translated is supposed to be one of the important aspects of the study of molecular biology. Apart from this, we have also studied the different tools of molecular biology, namely the various blotting techniques. We have also gone through the gel electrophoresis methodologies. We have discussed the PCR technology. And finally, to conclude, we have also emphasized on the importance of various molecular markers that help us to understand molecular biology in a better way.